Hello, everybody. Welcome to the Thermo Fisher Scientific Workshop titled Scalable AAV Manufacturing, Addressing Challenges Across the Workflow. My name is Natasha Luki. I'm a Senior Product Manager for Vial Delivery, and I'll be your moderator for today's workshop. Today, we have an exciting agenda with three great speakers that will present two of our solutions for AAV manufacturing, as well as provide you with an overview of the current cell and gene therapy regulatory landscape and virtual inspections. At Thermo Fisher, our mission is to enable our customers to make the world healthier, cleaner, and safer. At our core, we're scientists, and our purpose is to enable our customers to fulfill their scientific purpose. To us, the success of our customers is our success, and we take pride in fulfilling our mission. Cell and gene therapy is a dynamic market with rapidly evolving technologies and processes. We have built a foundation over decades of experience with biopharma using our products and services to support customers in this space. This foundation is rooted in quality products and a long-term commitment to the market. As the world leader in serving science, we have robust capabilities in gene therapy research and development. We're highly motivated to partner and collaborate with you to not only accelerate your clinical programs, but also to help advance the pace of development in this area as a whole. At Thermo Fisher, we have different solutions to meet your desired approach to AAV production. Our production system solutions with the AAV-MAX system provides you with a complete set of pre-optimized and regulatory compliant reagents that work together to maximize and simplify AAV production. If your interest is to optimize your own process, our standalone media library solution gives you the flexibility to obtain a customized media formulation geared toward optimizing your existing workflow. Lastly, through our PCR viral vector CDMO services, you can outsource your AAV manufacturing with confidence to a team of experts with years of experience and an unrivaled track record and capacity. For today's workshop, our speakers will be Xiaoyan Liu, Senior Manager of R&D in Cell Biology, Genius Stofa, R&D Staff Scientist in Molecular Biology, and Monica Comerford, Head of Regulatory Affairs for the Viral Vector Services. We will have a Q&A section following each presentation. If you have a question, please submit it by typing it in the chat box. And now let's move into our first presentation with Xiaoyan Liu, where she would talk about the AAV-MAX helper-free AAV production system. Xiaoyan, take it away.
Good afternoon, good evening. Thank you for everyone still stay on this time. As Natasha introduced, I'm going to talk about the AAV-MAX helper-free AAV production system. Thermo Fisher Scientific does offer end-to-end solutions that expand the entire viral vector workflow. We offer the products, services, expertise to help our customer to develop their breakthrough cell and gene therapies. In the AAV-MAX system, phase 2, the upstream process, highlighted here in the red. The system covered cell culture, cell expansion, plasmid transfection, and viral vector production. The AAV-MAX is a complete helper-free AAV production system based on the mammalian cell line. is Gabico brand scalable high titer system with simplified AAV production workflow. The prototype products was launched in the middle of February of this year. The RUO, the whole system, will be launched in the Q3 of 2021 this year with all validated performances. The Gabico brand cell therapy grade CTS AAV-MAX system will be launched the next year sometime. In this new system, we have the new cell line, media, transfection reagent kit, and complexation buffer, and the lysis buffer. I will address each component in this system in next slide. This system featured first with the new clonal 293F derived cell line we call the viral production cells 2.0, because we have the first generation VPC cells launched earlier with the lentivirus production system. Along with this new cell line, we have the amino original free, chemically defined, protein free, the new viral production media. This media not only support the cell culture expansion, as well as support the production. The third component in this system is the transfection reagent kit. So this kit includes the transfection reagent, include the transfection efficiency booster, and the AAV viral enhancer. All these reagents are chemical defined, amino original free. In this system, we developed a new complexation buffer, is LF chemical defined. The performance really equivalent to well-known OptiMEM, but help the customer easily to transit to all the ALF reagents. In addition to the cells, media, transfection reagents, we also include the Twin20-based lysis buffer for the convenient and the easy the downstream sample preparation. This system uses the typical triple transfection technology, basically 3 plasmid DNA, the gene of interest, RepCap plasmid, and the helper plasmid, and optimize the ratio and transfect it to the 293 producing cell line. And the viral caspase protein contain the gene of interest and formed. This slide show you the overall the transfection protocol. This is a small scale based on the shake flask scale, but it is very easy to transfer this protocol from this shake flask scale to the large bioreactor scale. Here I show the steps by step about this protocol. As you can see on the day 0 is the cell culture. Basically, this cell line gives you a robust performance. You do not have to do the day -1 subculture. You can take the cells from your routine culture. That means 0.6 milli per mil, culture 3 days. If you want short, you can do 1.2 milli for 2 days. And if you want to do the quick, fast transition from the small scale to large scale, you also can do 1.5 milli per mil, just culture 1 day. And on the day of transfection, you dilute the cells into the 3 milli with additional fresh media. That's typically for the first 2 conditions. And for the 1 day culture, the cells after 1 day, just about the 3 milli. You do not need to adjust the volume, adjust your larger scale parameters. And this moment, you can end our AAV-MAX enhancer. So if you forgot, is not a big deal. We have the robust flexibility in this protocol. You still can end your AAV-MAX enhancer post the transfection within 18 hours. And once the cells are prepared, and you can start to make your transfection complex. Typically, just like dilute DNA in 10% volume of the complexation buffer and relative to your culture, transfect culture volume. Once the DNA diluted, you can pre-mix our transfection reagents and the booster. This mix then add to diluted DNA and incubate at the room temperature about 20 minutes, then add to the cell culture. In here, I want to point out all the reagents you can pull out from the refrigerator directly. You do not need to warm up. You can directly use it. And after 3 days, you can harvest your crew culture and perform the AAV titer, make the lysate, or move to downstream purification, or depends your purpose. In this slide, I summarize some attributes of the new cell line, the VPC 2.0. It is a clonal cell line with approved clonality, is absence of SV40 large T antigen, and is high density suspension culture, and relatively give you a high AAV titer. The peak density of this cell line in our new viral production media is about 12 to 15 million per mil, based on the seeding density initially, and also the day you count the cells. The doubling time of this clone is about 23.5 hours. Based on the 3-day or 4-day culture, you calculate the doubling time. It's very good feature of this cell line is non-clumpy phenotype. If you have been using the 293 T cells, and you can really tell this cells is non-clumpy phenotype with robust scalability and great passage stability. The documented cGMP banked cells will be available together with our CTS version, the whole system. In this slide in the middle on the top, there are 3 images I want to show before the single cell cloning. After we ended the media to the 96 well plate, we imaged the well with clear bottom, clear the background. On the day of a single cell cloning, we catch the 1 cell, and the day after we catch 2 cells. The cells double. We continue to make an image, to take the image up to 2.5 weeks. And when the colony ready to be transferred from 96 well plate to 24 well plate, eventually transferred to shake flask for cell banking. On the bottom in the middle, I show you the growth curve analysis of the VPC 2.0 cells and the passage 12 and the passage 29. You can see almost exactly the growth pattern in the different passages. On the right panel on the top is doubling time. As I mentioned before, is average doubling time based on 3-day and 4 days culture. You can see the cells maintain the relatively steady doubling time over many passages. Another good feature I really want to point out here is the cells post the thaw, the quick recovery. If you compare with our first generation VPC, you would see the viability drop on the day post the thaw. But these cells, you do not really see any viability drop if you make the cell bank follow our protocol. This allows our customer to quickly get the cells expanded and to perform the viral production run. We apply the system approach to integrate each component in this system to achieve the optimal performance. In this slide, I show you the overall AAV genome titer measured by the qPCR. On the left panel, the red bar represents the 5 different serotypes we used and during our development. We use the GFP as a gene of interest in the development work. As you can see, across 5 different serotypes, we could get relatively 1 × 1011 VG per mil, the titer. In the middle panel is the viability of the VPC cells and the harvest about 3 days post the transfection. This viability shows much higher than our first generation VPC, almost 20% higher. The typical viability is about 75% based on the serotypes. Because we transfected the cells and relatively high density and 3 million per mil, the VCD, the viable cell density and harvest is about 4.5 to 5 million per mil. How about this system? Can you use this system to the large scale? How stable our transfection reagents with together the DNA to form the complex? On the left side, I show 2 different serotypes. If we premix our transfection reagents and the booster, how stable they are. And you can see less than 1 minute up to hours and days. This premixed reagents is quite stable. Lately, we tested about 1 month. This mix still perform as good as freshly mixed. On the right side, I show you the complex. Once the transfection reagent and booster mix together with the DNA, how stable they are, how fast you can use. So definitely less than 1 minute, the complex didn't form. And you need at least 10 minutes. but about 20 to 30 minutes is relatively stable around that time. When we developed this system, we were asked each dilution step needed to be at least 10 minutes, and the complexation time needed to be 20 to 30 minutes, able to allow the customer to use this system at a large scale. How about the scalability of this system? We tested each size of the shake flask from 125 mils to the 5 liter with the final volume of about 2.2 liter in the 5 liter shake flask. As you can see, across different scales, and the relative, the AAV to titer is really comparable. And also we use the H10NT cells to do the infectivity assay. And we use the virus generated with this system and the different scale, infected the 1080 cell line and with the serial dilution and check with the flow cytometry. On the right panel, we show the bioreactor. It is a finesse and stir tank bioreactor. The capacity is about 3 liter. The final working volume is about 2 liter. As you can see, the relative, the VG per mil, the titer is really comparable between the shake flask and the bioreactor. In addition, the growth profile in these two different type of the culture vessel and relatively really comparable. How about the repeatability and the reproducibility of this system? As I mentioned during the development, we tested different serotypes. We run different time with different operator and different experiment. As everybody knows, the qPCR assay has its own very variable level there. And I would say based on the cells, the performance, each run, and even different operator, the cell viability and the cell VCD and the time of the harvest relative consistent. So this variation not only just cells performance, we believe a majority is the assay itself to bring some variation for this system. How about the comparison study with our competitors' reagents? Typically, the PI Pro, PI-MAX, these two transfection reagents, well known and well used, used in the AAV production field. So, and the red bar you can see represent our whole AAV system with the cells, media, transfection reagent, and the AAV titer enhancer. And compared the VPC 2.0 cultured in the viral production media use different transfection reagents. We also compared with our AAV-MAX system. Although this system is developed for the lentivirus production, but we learned a lot of customers start to use this system also for the AAV production. As you can see, our AAV production system is auto-performed to other transfection region or other systems. As I mentioned, this is the new version and viral production cell line 2.0, how to compare with our first generation VPCs. Here I compared this just like apple to apple comparison, the truly cell line comparison. So I adapted the VPC cells to the new viral production media for 5 passages, then use exactly same transfection protocol, transfection reagents with the AAV-MAX system, just compare the performance between these 2 cell lines. As you can see, almost 5 to 10 folds the titer increase with the new cell line. On the right side is a culture media comparison. We know a lot of customers use Gibico Freestyle 17 media as the AAV production media, and also the Irvine Balance CD HAC 293 media also support the 293 cell growth. And we compare our whole system with this 2 media in combination with the PI Pro. And this tested with 2 different serotypes. On the left, the group is AAV2, on the right is AAV6. Finally, I want to give some the information about the purification protocol. If you use our AAV-MAX system and what's the typical purification process, This is a Schick flux scale, about 200 to 300 mils, the purification protocol. We use the GFP AAV-6 as an example. So here we listed on the day 3. Day 3 means if you consider the protocol I showed you before, day 3 is the harvest time. You harvest the crude culture and our lysis buffer and to the 1X and the spike with some chloride mechanism to the lysate and go through with the benzonase treatment and then move forward to clarification step and the DE to the lysate and filter and centrifuge, finally collect the clarified lysate and use a thermofacial gold pure AAV-MAX, the resin, to do the purification. For the purification buffers, I'm not going to read line by line. I just want to show you some feature of the purified AAV particles from this AAV-MAX system. As you can see here, we loaded 200 mils and the elution with the 5 mils. This shock peak of the fraction and represent the robust purity and about 70% were recovered the AAV-6 particles. And furthermore, we run this purified fraction of the AAV-6 on the SDS-PH gel with the chromosome blue stain. As you can see, the caspase protein, the VP1, VP2, VP3 is the elution fraction and shown in this gel. On the right side is the transmission electron microscope. The image shows the consistent of the mixture of the empty and the full particles. And we calculated the full particle is about 32% and the empty is about 68%. And in the summary, the AAV-MAX system is a scalable high titer system. And we are in the process to do furthermore the downstream purification, characterization, and analytics. And thank you very much for your attention and happy to address any question you might have.
Thank you for the insightful presentation. We'll now start our Q&A session for this first presentation. As a reminder, if you have a question, you can type it in the chat box and we'll address as many questions as we can. So to kick things off, our first question is, what is the largest scale and highest density you have tested the cell line, Qiaoyan?
The scales here we tested, I show you the data is about 5 liter Schick flux and the bioreactor 3 liter. So our collaborator is testing the 50 liters now. And we can show you that data very soon. So for the density, we tested 2 million, 3 million, 4 million, up to 5 million. And the robust level, we suggested to use 3 million.
Thank you, Qiaoyan. Our next question is, how different is the LV-MAX enhancer and the AAV-MAX enhancer?
This definitely we cannot tell you what the enhancer real is. And I can tell you the enhancer in the AAV-MAX is a complex cocktail. In this AAV-MAX enhancer is the LF chemical defined, is really, good reagents, help you move forward, for the further CTS version. Less, the regulatory concern.
Thank you, Qiaoyan. Our next question is, how do VPC's 2.0 cells differ from the LV-MAX VPCs?
Oh, LV-MAX VPC is adapted cell pool. It's not a single cell clone. And is the pool cells just adapted in the rich media and increase the performance. And this VPC 2.0, as I mentioned, is a true clonal cell line with clearly and the lineage documentation and prove the clonality. Thank
you, Qiaoyan. And there is a follow-up question to that. how was the level, if you measured, how was the level of HCD and HCP compared to 293Ts?
293T, to be honest, we are just in the process to start to compare with 293T. I do not really have that data right now and to give that answer. Sorry about that.
No problem. Let's move on to the next question. The question is there a GMP version of the cell line?
Yeah, as I mentioned, we are in the process to make the cGMP bank, the cell line, will be available next year. I believe probably the second quarter. That's what we aim on.
Thank you, Xiaoyan. Our next question is, has the AAV-MAX transfection reagent been accepted by the FDA? If FDA requires material or components information and toxicity data, could you provide? It's a long question.
I believe so. That's what we work hard to have the CTS version AAV-MAX system. That means when that system complete, we have the whole thing, the whole system, each component in that system should be just like less regulatory concern. That's the purpose we have the cell therapy grade reagents.
Thank you, Xiaoyan. Our next question is, will the components be available separately or they will be sold only as a kit?
I could predict we are going to facing these questions. And as right now, I believe the media standalone, cell line can be purchased separately too, and the transfection reagent will be sold as a kit. That means include the transfection reagent, booster, and the enhancer. These three will form the kit, and complexation buffer can be separate. But although you can purchase the different, the reagents separately, I would suggest, as the whole system, if you want to reach optimal performance, is the whole system optimized, on this level, I still suggest you better to use the whole system. Thank you.
Thank you, Xiaoyan. Our next question is, can VPC's 2.0 cell line be used with the AAV-MAX system?
Yes, the answer is yes. We just tested and better than VPC. That's one thing I can tell. But we need to do more work. We just started. That's based on the customer request. They do ask the cells can be used for every, yes.
Okay. Our next question is, why do you call the AAV-MAX system a helper-free system?
Oh, that's related to the triple transfection technology. That's eliminate the wild type adenovirus to help to do the infection. And we do not need any alive virus. We just use adenovirus derived plasmid DNA as a helper DNA to do the triple transfection. That's why we call it helper-free. That means alive the virus helper-free.
Thank you, Xiaoyan. Our next question is, you showed a media comparison, but it didn't include AAV-MAX medium. Have you compared the AAV-MAX medium to AAV-MAX medium?
We didn't do that on purpose, but I do believe that media, the performance between the AAV-MAX media and the F17.
Okay, thank you, Xiaoyan. Our next question is, are there or will there be residual reagent kits for the components of the AAV-MAX system?
I do believe our Thermo Fisher has a broad range of catalog products to help to do downstream analytic, just like a whole cell DNA, residual DNA, the kit. And if you really need this information, we definitely can help to, to provide.
Mm-hmm. I believe the question is if we are going to have residual kits for the transfection reagent within the AAV-MAX system.
currently is not in our group. We didn't plan to develop that. I think across the Thermo Fisher, we should have.
Thank you, Xiaoyan. Our next question, thank you very much for the questions, by the way. We're getting a lot of questions. What are the max VG per cell that was achieved with the AAV-MAX system?
Oh, if we can quick, very easy to do that calculation, because we start with 3 million per mil, the cells, and we can use that cell number to calculate the VG per mil. Once the cells produce the AAV, we know the cells, the viability goes down, the cells die, and the AAV viral particle released to the supernatant. So we can simply, calculate based on the initial cell number. And that's just like a different serotype based specific. Yeah, that's not a really challenging thing. We can quickly to do the calculation. But we just follow typical, the VG per mil, this kind of a way to show the performance. So we didn't really show, the VG per cell, but we definitely can calculate, no problem.
Thank you, Xiaoyan. Our next and last question is, why are AAV2 and AAV6 normally used here to test the system as opposed to other serotypes like AAV9 or AAV8?
Oh, that's related to our purification, the resin. We established the purification, lab, and we start with this 2 resin and just like get more experience. So we are going to do all serotypes of purification. We will provide more information, get along, yeah.
Thank you, Xiaoyan, very much for answering all the questions. We are out of time. Thank you, everybody, for submitting your questions. So we're now going to move to our next speaker, Gino Stoffa, and his presentation titled Viral Vector HEC Media Panel. Gino, take it away.
Going, everybody. As a complementary approach to the AAV-MAX kit, we've developed a viral vector HEC media panel to help our customers optimize their own process and using their own cell lines to achieve higher productivity. And the reason why we want to use a panel is to address the diversity out there that is derived from the many different HEC cell lineages that our customers are using. So these HEC cell lineages can differ in gene expression profiles as well as metabolic needs. And what you can see on the right here is that these different lineages cluster differently in a PCA analysis, indicating that they express their genes differently. So we've developed a HEC media panel with 5 chemically defined formulations that allow you to rapidly test for improved titer. They're compatible with a helper-free triple transfection, and they come with a personalized consultation from our field application scientists. Now, using these diverse media, you can screen you can rapidly screen the library that goes beyond our catalog offerings. And these diverse formulations will hopefully be able to capture the diversity of the unique HEC cell lines and processes that you have, allowing you to get a final medium that will let you have improved titer. Now to do this, we initially performed principal component analysis on 58 internal media formulations and narrowed it down to the 17 of the most diverse ones that we could find. Out of those, we screened them with 2 different lineages of HEC293 cells for growth, transfection efficiency, and titer. And from there, we narrowed them down to a panel of 5 diverse media, which the principal components analysis on the left there gives you an idea of how diverse they are along the 2 main principal component axes. And the media panel heat map on the right gives you an idea of how diverse they are in their different types of nutrients that they provide. Now, to further assess these media, or to confirm our selection of the media, we tested the media using 3 HEC293 clones with 3 different AAV production processes to produce AAV2. And using PEI Pro as the transfection reagent. Each of these different clones had a different process and different growth parameters. They represent 2 different hex cell lineages. 2 of them come from 293F, and one of them is a 293S lineage. And they're supplemented differently with either glutamax or glutamine, and they use different seeding densities. They were also banked in different media. And they were all able to be adapted to the panel. The 293F clones were able to adapt directly, whereas the 293S clone required some sequential adaptation to get them in the media. So we tested, as I mentioned before, we tested these different cell lines in a transfection process to produce AAV2. These were all shake flask experiments at the 30 milliliter scale. So we transfected them normally. They all had different seeding densities, either 3 million cells per milliliter on day 1 or 3, or 1 million cells per milliliter. And the cells were transfected and harvested 72 hours post-transfection. There's no other enhancers used here. It's just the media that is giving us the result. But we did add glucose back post-transfection up to 6 grams per liter if it was necessary. The DNA concentration was 1.5 micrograms per milliliter for the 293F cells and 1 microgram per milliliter for the 293S. And the ratio of PEI to DNA was dependent on the media that we used for the most part. And we used also different ratios of the GFP transfer construct, the rep cap, and the helper plasmids. During the transfection process, if you look at cell growth, you can see that the panel media doesn't actually differ too much from the F17 control medium here, indicating that you don't really have to change your growth parameters if you wanted to use these media on your own cell line. So it's kind of a plug and play approach. I know the 293S didn't do too well with panel 2 here, but for the most part, the cell growth is very similar among the panel media compared to control. Transfection efficiency varied among the clones, but we got anywhere between 30 to 70% transfection efficiency. There's no systematic correlation to AAV titer from this. And these media are amenable to be used as a complexation buffer, or you can use the complexation buffer of your choice to do these transfections. So it's basically you keep your own protocol. If we look at AAV titer, we can see that the titer values differ and are dependent on the HEC293 cell type, or cell line or clone. And you can see that in the first 293F clone, that panels 4 and 5 are about a 10-fold increase over F17, whereas in the second 293F clone, we have about a 2-fold increase over F17. In the 293S cells, you can see that the panel didn't perform as well as F17, but panels 4 and 5 were similar in growth, or similar in titer to the control there. So this indicates that what you get from the media is really dependent on the cell line, and we're capturing the diversity of different cells or HEC293 clones that are out there. So the viral vector media panel consists of all AOF formulations to level 2, so animal origin free. There are no growth factors or proteins in the panel, and they are scalable to cGMP scale as liquids or our GIBCO AGT dry format. And you can either use glutamine or Glutamax at whatever concentration that your cell line requires. So in summary here, we have 5 formulations, a HEC293 panel of 5 formulations that cover a wide range of components and concentrations that allow us to capture the diversity of all the HEC293 clones and processes out there. They provide comparable growth to F17 control media. And the effect of the panel medium on AAV production is dependent on the HEC293 clone and process. Yeah, the viral vector media panel addresses the diversity of HEC293 clones and AAV production processes. And with that, I can take any questions.
Thank you, Gino, for that informative presentation. We'll now move into our Q&A session for this presentation. As a reminder, if you have a question for Gino, please type it in the chat and we'll get to as many questions as we can. Our first question is, did you control for pH in flask screens by feeding sodium carbonate, or was this not necessary for your process?
So no, we didn't control for pH during the flasks, and while we didn't measure the pH, it's unlikely that it was necessary since the cell densities were relatively low.
Thank you, Gino. Our next question is, any thoughts on how using PEI-based transfection versus liposuction might impact media selection?
So we didn't actually test any lipid-mediated transfection here. We only used the polycationic PEI Pro in this case. But we do think that these media should be amenable to both. And where the media, or really your results are likely more dependent on what your cell line and process is, as opposed to what transfection reagent you're going to use with these.
Mm-hmm. There was a follow-up question to that question asking about testing liposuction-based adjuvants, but I assume you haven't tested.
No, we have not tested any of those.
Okay. Our next question is, did you evaluate the impact of those media to the downstream process?
Unfortunately, we haven't yet, but we do plan on doing that work in the future.
Okay. Our next question is, are those panel media already commercially available?
So yeah, I guess I should have mentioned this earlier, but the panel media launch, or the panel launched at the end of March, and they are available. I think, yeah, they are available. I forget exactly how you go about ordering them, but they are available.
Oh, good. I'm sure they can find it easily on our website. Our next question is, do you offer any nutrient feeds for enhancing the cell densities, and did you test them in any way?
So we haven't tested any feeds with this process either. though that is something we plan on doing in the future. And these media should be amenable to any feeds that you're currently using as well. So whatever your process is, if you do have feeds or you have any type of supplements, this media should work with them.
Thank you, Gina. Our next question is, in addition to the HEC lineage, what other factors could affect the media selection?
So other than the HEC lineage, what your actual process would be. So, what cell density you're actually growing these to before you transfect, or, when you transfect, when you harvest, what your reactor settings are and all that stuff. Yeah, but like I said, we only tested in shake flasks and at small scale for this work.
Thank you, Gina. Our next question is, are the 5 media all designed to be both complexation and culture media?
Yeah, so all the media are amenable to complexation as well as culturing. So you can complex in any of these and they should work fine. There's nothing in them that would prevent that.
Thank you. Our next question is each of the media panel available at GMP grade with a drug master file?
I'll have to get back to you on the drug master file portion. I don't know about that, but they are scalable to GMP. You would have to go through our custom orders process to scale them up with any of our panels. I think that's something, I'll get back to you on the drug master file portion. So sorry, I don't have the answer here.
Next question is, have you compared this media to the XB293 expression medium?
We haven't compared it directly to XB293 expression media. Most of our comparisons were done to F17 here, and then the other internal media formulations that we had tested.
Thank you, Gina. Our next question is, what is the maximum volumetric titer by using the panel media?
So the maximum titer that we got using the panel media was around 1E11 VG per milliliter. And we only looked at VG per milliliter. We did not do any other type of titration or titering.
Thank you. All right, the questions keep coming in, Gina. I hope you're ready. Our next question is, can you share if you're working on high cell density process intensification media for AAV production?
So currently we're not, we didn't test anything at high density, but I mentioned before that we are, we will look at higher density in the future. We are looking at feeds and other stuff like that to be amenable to higher density culture.
Thank you. Our next question is, do you think this media panel help improve the performance of VPCs or VPCs 2.0 cell lines?
I'm not sure. We haven't tested them with VPCs at all. We just used our own internal 293F clones here and the 293S that I mentioned that was from the National Research Council of Canada. That's something we just haven't tried.
Thank you, Gina. Our next question is, can you also use this media for lentivirus production?
So we haven't tested it for lentivirus production. It's possible that it could work. It does grow the hex cells fine. But yeah, we haven't tested it. That's all I have, sorry.
No problem. I actually have a question myself, so I'm going to introduce myself here and ask. I'm curious to know if you've looked at adapting adherent cell lines to your panel media.
So we haven't adapted the cell lines in-house, but we do have a collaborator that was doing that work for us, and they were able to adapt 293T cells to some subset of the panel media. And only because they only tested a subset because they only had so much time. But yeah, the adherent cells are you can adapt adherent cells to suspension in the media.
Okay, thank you, Gino. It looks like we don't have any more questions in the chat. If you want to ask a last question, please go ahead and submit it right away before we move to our next speaker. We have about 1 minute for one last question for Gino, if you have it. If no more questions, we can go ahead and move to our last speaker of the workshop, Monica Comerford. And Monica will be speaking about virtual inspections, navigating the new paradigm. So Monica, please take it away.
Good evening, everyone. It is an absolute pleasure for me to be here today to present at ASCGT 2021. As Natasha indicated, my name is Monica Comerford, and I'm the head of regulatory affairs at Thermo Fisher Scientific Patheon Viral Vector Services, formerly Brammer Bio. Now, prior to joining Thermo Fisher Scientific, I was a product quality microbiology reviewer and pre-licensing inspection lead for BLAs in the Office of Pharmaceutical Quality in FDA's Center for Drugs Evaluation and Research. So really understanding the regulatory landscape of biologics has been a big part of my life now for more than 5 years. Now, those were fantastic presentations from Yao Chan and Gino, and they were quite scientifically based. However, for the next 20 minutes, we'll shift gears slightly as I present to you a case study in how we at Thermo Fisher Scientific are using cutting-edge technology to implement virtual inspections to enable us to perform our mission-critical regulatory commitments. Now, it's been an exciting past few years for us at Brammer Bio, and we're privileged to be presenting here as Thermo Fisher Scientific Patheon Viral Vector Services. Now, as we are celebrating 2 years since the acquisition of Brammer Bio by Thermo Fisher Scientific, we continue to see the huge potential and opportunities that this brings in order to allow us to provide critical therapies to the people who need them, the patients. And we're all thrilled to be a part of Thermo Fisher Scientific's efforts and to bring their expertise to help us bring these products to market faster. Now, just to give you a little bit of history before we start talking about the new technology that we've been implementing across our network, I wanted to make you aware of how much history we have as a cell and gene therapy manufacturer. Now, Brammer Bio was founded by Richard Snyder back in 2006 with a vision to make vectors for clinical investigation and for clients. In 2016, Mark Bamforth merged Florida Biologics with Brammer Bio, and following this, we've grown from 100 employees to over 1,000, and I think that's pretty impressive. Now, this led us to ultimately being acquired by Thermo Fisher Scientific in May of 2019. Our North American sites consist of 4 sites: the founding site in Alachua, Florida, which is our early phase clinical development site, our 2 late phase commercial sites in Cambridge and Lexington, and our GMP warehouse in Somerville. Now, because we are committed to growth, we have publicly announced plans to build a new 290,000 square foot commercial manufacturing facility in Plainville, Massachusetts, which we expect to be operational in 2022. Now, as we all know, viral vectors are modified viruses that we use as tools to deliver specific genetic material into cells and gene modified cell therapies. At Patheon Viral Vector Services, or VVS for short, we focus on viral vector gene therapies where there is an in vivo delivery of specific viral vectors into a patient's cells in order to treat a specific disease. Now, on the other hand, as we all know, gene-modified cell therapies or ex vivo therapies where these patients' cells are collected and modified using viral vector gene therapies, and then therapies are then provided back to the patient. Now, there are many unique attributes for the cell and gene therapy sector, as I'm sure we've heard throughout this entire conference today. First is the urgency to bring these products to market faster because these therapies have a potential to have a huge and dramatic impact on patients' lives. Now, because of this, regulators are providing formal and informal guidance due to the need for these products to get to market faster. Now, these treatments are also particularly unique because of the market dynamics, since you are really looking to make cures for patients rather than treating patients with long-term treatments. And as you can see on the graph here on the right, initially there's a high bolus of patients being treated, and then that rate really drops to the rate of incidence as compared to the traditional monoclonal antibody treatments that we may be used to, which really in effect treats patients long-term. And even though there are unique pros and cons to this therapy, the cell and gene therapy universe is really exploding right now because of a common mission that we have, which is the rush to find cures for patients. And if we look at this graph it's a little outdated from 2018, but there's some really explosive growth in this field. So for example, there are currently 512 companies that are developing cell and gene therapies in the United States alone, according to the Alliance for Regenerative Medicine. And while it may take 10 years to bring a product to market, cell and gene therapies have the potential to cure patients. And really, this type of idea is a paradigm shift from the standard long-term treatment therapies that we talked about on the previous slide. And like I said, it's happening because we are all passionate about the benefits this would provide to patients. And of course, the industry isn't doing this alone in isolation. We're working together with regulators to grow our understanding. Now, regulators had certainly had challenges this past year as they are dealing with the COVID-19 pandemic. And in fact, both CDER and CBER, so the Center for Drugs Evaluation and Research and the Center for Biologics Evaluation and Research, indicated that there's an increase in their workloads that are related to the SARS-CoV-2 crisis. And it has been challenging to meet not only target meeting dates, but the FDA has also delayed approval of marketing applications due to delayed inspections. However, the technology that I'll be talking about today will help enable regulators to meet their regulatory commitments and timelines, and in turn help us help them. Now, with that background, I really want to focus on the technology that put our sites on the forefront of supporting our clients and also supporting their regulatory submissions to regulatory health authorities. And I particularly wanted to touch upon this topic as we are living during a time at which we are trying to use every resource at our disposal to maintain business continuity. Now, across all of our sites, we have been implementing virtual inspection technology to support both client audits and inspections from regulatory health authorities. And with these sites in our network, we wanted to ensure there was a way to enable business and regulatory continuity during this challenging time. But before we talk about how we implemented these virtual inspection tools at our sites, I first wanted to give you an overview of what the regulatory landscape looks like in terms of virtual inspections, and also to give you a flavor of why virtual inspections may be a much needed resource here, especially where business continuity needs to be managed when travel restrictions are in place. Then I'd like to hit on the advantages and disadvantages that virtual inspections provide over the traditional on-site inspections and how we've been able to implement it at our VVS sites. And then I'll end with what we think the future state of virtual inspections would be in the coming years. Now, as we all know, and as I've alluded to, the world has been living in a pandemic for well over a year now. And in that time, where the biopharmaceutical industry has become global, there are many challenges that need to be overcome in order to ensure that the people we care about most, the patients, are getting the medicines that they need. One major challenge is how do we allow regulators to keep their commitments to the public by performing necessary inspections? And as we all know, these inspections are a critical part of the pharmaceutical and biotech manufacturing global infrastructure because they are needed to verify that manufacturers are indeed manufacturing under CGMPs to ensure that drugs are safe, pure, and potent. Now, during this COVID-19 crisis, both the US FDA and the EMA have scaled back regulatory inspections. The FDA recently published the Resiliency Roadmap for FDA inspection oversight a week or two ago, where they indicated that they received nearly 13,500 applications within the past year, and approximately 600 of those needed inspections. And regulatory authorities like the Australian Therapeutic Goods Administration, the UK's MHRA, the EMA, and Russia's State Institute of Drugs and Goods have all performed virtual inspections. And because of its promise and ability to overcome the traditional disadvantages that an on-site inspection can have, like how do you travel during a travel ban? A lot of pharmaceutical groups have reached out to the FDA and suggested that virtual inspections are absolutely needed during this crisis. And while it may now be outdated because the FDA recently published a guidance for virtual inspections at the end of April, I wanted to just call out this Fierce Pharma article that indicates that virtual facility inspections during a pandemic can help ensure the continuous supply of much needed drugs. Now, this article was published last year at the height of the pandemic. But the continuous supply of these much needed drugs is not just needed for the US, but it's needed worldwide. Now, as I've alluded to, the FDA has only recently published in April a guidance on remote interactive evaluations of drug manufacturing establishments, where they provide an overview of what the virtual or remote inspection program looks like. Now, recently, Alonzo Cruz at the FDA ORA speculated, and I'm kind of paraphrasing here, that there really is no going back from virtual inspections. It's now in the FDA's toolkit, and seeing how really the question is, how is the agency going to incorporate it into its oversight is really where the agency is going. Now, these are only recent statements by the FDA, but this cutting edge technology has been in the works for some time at Thermo Fisher Scientific. Because of our commitment to our patients and our pharma services Tech Ops Group team, they really led the way for the use of mixed reality platforms enterprise-wide, and had already developed a path for VVS to use virtual inspections and the virtual inspection experience for client audits and regulatory health authority inspections. And this certainly has been timely for us, as this path has allowed us to maintain business continuity without sacrificing client and regulatory responsibilities. Now, as part of our virtual inspection option, we can use Microsoft Teams or software that they're already comfortable with using in our daily activities. And there are features of Teams that allow us to have a review of quality documents during the inspection real time, with also real-time video conferencing for discussion. In addition, we have real-time online video streaming tours of our GMP facilities using a 360-degree camera. Now, on the right, you can see an image of one of our staff interacting and talking with an EMA inspector in real time, as if the inspector is in the room with her during our Alachua, Florida EMA inspection in May of 2020. Now, I actually love this photo because at the bottom here, you see our head of quality at our Alachua site, Tony DeFusco. It's not the inspector that came. And what you can see, though, is he's intently listening to this interaction. But what's wonderful about it is he has an opportunity to chime in if there's any question that the subject matter expert is not able to answer. And this really allows for an interactive experience for our inspector and our teams. Now, how do we perform this live stream tour, and what other technology do we use? As you can see in these pictures, we have multiple pieces of technology, including a 360-degree camera with AVATour software, the Microsoft HoloLens, and Matterport 3D mapping. Now, using the 360-degree camera isn't as daunting as you would think. For our setup, we've attached the camera to a cell phone or an iPad and mounted it on an IV pole. Now, this pole can be wheeled around the facility on smooth surfaces, and the camera itself has high enough pixelation to read documents, labels, and logbooks during the tour. Now, the other advantage is its cleanability. And if you need, you can go into and out of rooms with the appropriate cleaning studies performed, of course. Another piece of technology that can be used is the Microsoft HoloLens, and it's a virtual reality piece of technology that allows us to perform activities that we may not otherwise show on the 360-degree camera. Finally, we have that Matterport 3D mapping technology. Now on this next slide, I'll show you a video of how that works. Just give it a moment to start for us. Now, as you can see here, as we are going through the facility, we have a video of what the Matterport 3D mapping of our QC lab space and lateral looks like. Going through these scans is very similar as going through scans of a virtual home tour on websites like Zillow or Redfin. And as you can see, as we're going through the video, you can see the space in high resolution and you get 360-degree views of the space as well. Areas of the space are clickable and thus viewable. And honestly, when you look at the scans, I sometimes wonder if their rendering's too good because you can see how beautiful, sunny the Florida weather is right outside of the QC labs. But now the virtual inspection technology is also, and its capabilities are only one part of the inspectional experience. And it's really paired with the second element of the inspection, which is the document review and control of document review, or really the platform that you use to host the inspection. Using Microsoft Teams can help to organize the documents in a way that facilitates review by inspectors and allows us to store them in a controlled manner. Now on this slide, I just have a simple example of how you can organize your documents into directories and folders within Teams, and this can be provided to inspectors as well. Now, while Teams can be viewed as a native application on your computer, it can also be solely viewed on the web, and viewing it as a web-based browser does have its advantages for both sides because one, we have more control of the documents on our side, and two, we actually found that the EMA was accepting of viewing these files temporarily on the web because they have their own infrastructure to ensure that their computers and infrastructure was not compromised. Now, switching gears slightly, I really wanted to think about the roles performed during the virtual inspection. First, we found that the same roles typically apply between on-site inspections versus virtual ones. And you still have your escorts, your scribes, your ready room, your strategy room. But we found that one component now has more time in the spotlight, and this is digital support. This group became key in order to help us ensure a smooth inspectional experience. And we also found that with this cutting edge technology, our IT department was very excited about it because they were able to showcase their IT skill. And honestly, we were able to highlight it, which made us very excited for our teams. And in general, all these roles that we see on the slide are happening digitally, just within different channels within Microsoft Teams. And controlled access is important here. And it's similar to the controlled access that you would have if you were in an on-site inspection as well. Now, in order to ensure a smooth inspectional experience, it would also require you to perform wireless access point data mapping or wireless heat mapping. Now, we did this at our site to understand the wireless signal strength in all of our rooms at our facility so the cell phone or the iPad can work appropriately, but our site had already had a long history of cell phone use in our rooms, so signal strength wasn't a concern for us. And based on our experience, we would recommend having multiple 360-degree cameras ready and staged throughout the site in places you know you'll be touring or the inspectors will want to view. This helps to reduce cleaning and transition time so there's no dead air during the tour. And finally, we would recommend using 5G as it lends credibility for the inspection. So for example, when the power goes out, the inspectors will be able to see what happens and what goes on, so the inspection doesn't go dark. Now, another way to ensure a smooth inspectional experience is training. On the facility side, performing mock virtual inspection runs with your team is very useful so they know what to expect. And if the staff will be performing any activity using the HoloLens or showing a space that's not accessible by the camera, you do want to mock that ahead of time. So that way your staff can understand what it's like to be in that virtual reality environment. Now, there is a lot of work upfront to help ensure that the experience for both your teams and the inspectors is smooth, but you will find that there will be room for improvement and potentially more trainings would be needed to flush out any additional issues that you may be experiencing during the mock inspections. Now, one other item to consider is having a pre-meeting with inspectors or client auditors to really help you gauge their level of understanding of the software. Do they need more help or less help managing the platform? These pre-meetings also help you to build credibility for the platform so they can see that, yes, this is like an on-site inspection. And it's very important to help ensure that this is a good and trusted experience for your inspector. This also actually gives you the opportunity to meet with them ahead of time without the pressure of being in an inspection in order to build that rapport that you may somewhat lack when you have these virtual meetings. And you can also discuss pre-meeting requests and schedules to ensure that the inspection ends on time. Now, there are some advantages and disadvantages to both an on-site inspection and a virtual one, but I really want to briefly touch upon the differences we observed with conducting an inspection virtually. We do find that they both take up a similar amount of time, the pre-meeting notwithstanding. And on our EMA inspection in May, we did have 4 half days rather than 2 full days of the inspection. But this really worked to our advantage because it allowed us to maintain work during business hours and really use the other 4 hours to prep any questions that the inspectors had. Now, we also found the inspectors typically like to stick together, so this led to a slower dynamic, and it really helped us to better understand what the needs of the inspectors were, which also kind of led to a lot more questions. Now, we did also hold our inspection over the course of non-consecutive days, and we found that this really eased the burden of hosting responsibilities for the site. And we did notice that during this time, the questions that were posed during normal discourse are typically answered during tours. That dynamic had somewhat changed during the virtual inspection, and now these questions became part of a list the inspectors provided to us. Now, the really big advantage here is the time to prepare the responses and to present, and also the fact that SMEs from multiple sites can participate and be able to answer questions live without traveling. They're available during the tours, during the document review, et cetera, and we found this to be a big win for us. Also, you can't really read body language, so it's sometimes hard to get a read on the inspectors during the inspection, so what we recommend is really doing a frequent check-in with them. Now, while there is prep work associated with using the software and the technology, it really is needed for the experience, but once it's in place, it becomes standard and really maintained like you would for a traditional inspection. And a lot of these advantages are simply due to the fact that this platform is really in its infancy. And as we use it more, we grow our understanding and knowledge of it, so it will become standard for us. Now that we've concluded our EMA inspection at our Alachua, Florida site, like I said, at the end of May 2020, we've used this technology numerous times since then for client audits all across the site. Our experience has shown us that this is a viable platform that will revolutionize how we perform inspections, and really the technology hasn't gone unnoticed by the regulatory authorities. In fact, the EMA inspector had this to say, and quote, "This has been a new thing for us and has been a positive experience, but you may not realize it, but I have this on a bigger screen and the audit is almost life-size." Now, there are struggles with inspections and auditing, but there have been solutions to those. And what success looked like to us is that the auditors can adequately assess the environment, have access to our procedures and SOPs, and to our SMEs, and regardless of the inspectional outcome. And during our EMA inspection, we did get some minor observations, but we view this as validating our platform, indicating that it's just as good, if not better, than a traditional inspection. And in fact, during the end of the meeting at our closeout, the inspector used the phrase "during the tour," and it wasn't qualified with virtual. And this showed us that really the platform had been accepted as comparable to a traditional on-site inspection. Now, this I really think is a turning point for us here, because we can use this technology now and also in the future to help maintain business continuity. With that, I know I'm running over a little bit on time, so I wanted to acknowledge the people that helped me put this information together for you today. And I am now happy to take any questions that you may have, and I wanted to thank you for your time.
Thank you, Monica, for that great presentation. We'll now move into the Q&A portion. If you have any questions, as a reminder, please type them in the chat box and we'll get to as many questions as we can. Monica, our first question is, what are some of the regulatory challenges of applying lentivirus vectors to deliver therapeutic genes in vivo as opposed to ex vivo?
You know, I think that's a really good question. And in terms of regulatory challenges, I think, you know, it's a hard one for me to answer, but I really think that it comes down to what the expectation of regulatory authorities are. I don't necessarily want to elaborate on that right now, and maybe we can move on to the next question because I don't think I have a good answer.
No problem. I actually have a question myself, if that's okay. I'm going to jump in here. But you spoke very greatly about the virtual inspections. They seem very intriguing. I wonder, what are your thoughts about using virtual inspections after the pandemic? Do you think they're here to stay?
Oh yeah, of course. I think, like I said, Alonzo Cruz at the ORA at the FDA had said that it's now in their toolkit. They're likely going to use this resource. It's just how are they going to use this resource? Even the UK's MHRA has already announced that they're potentially considering hybrid approaches in the future where based on the risk or compliance history of the site, there may be an opportunity to perform a virtual inspection rather than an on-site inspection. And as we've seen with this pandemic recently, whenever an issue arises or another pandemic potentially arises, having these tools available to us now and harmonizing these tools in the future will really help us to maintain that business continuity, like I had mentioned.
Thank you, Monica. Another question is, what criteria can be applied to determining whether a virtual inspection can take place?
You know, that's also a really good question. And I think that really the FDA did discuss this briefly in their recent guidance document on remote and virtual inspection assessments. And the way I really think about it, and I kind of touched upon this in my previous answer, is where you have to think about the compliance history of the site and perform your own risk assessment on whether it's appropriate to perform a virtual inspection. So let me give you an example. If a site does not have a compliance history because they're brand new at manufacturing, it's likely that the FDA or a regulatory authority is going to want to come on site because they don't have that trusted experience with you. However, if you've had a long inspectional history of NAI or potentially VAI with the regulators, there may be an opportunity there that you will get a virtual inspection. Another opportunity is where perhaps you just had a surveillance inspection or a pre-licensing inspection. The likelihood of another inspection coming, it may potentially be higher, but it also may be in the form of virtual inspection rather than an on-site inspection because you recently had one. I hope that makes sense and answers the question.
I think so. If not, whoever posed that question can answer, can get another question in.
There we go.
All right, so I think what we're gonna do, please continue to submit your questions to Monica. We have about 5 minutes left. What we would like to do is open up the Q&A session to all three speakers. So if you have a question for Xiaoyan about AAV-MAX system, for Gino on the HEC Media panel, or Monica on the regulatory landscape or virtual inspections, please submit your questions via chat and we'll get to as many questions as we can. We'll start with one question, which I believe is posed to both you, Xiaoyan, and Gino. So I'll ask the question and Xiaoyan, you can provide your answer first, followed by Gino. So the question is, what was the highest VG per mil level that you've seen with the AAV-MAX system, Xiaoyan?
We could say about 3 × 10^11.
Thank you. Same question to you, Gino. What is the highest VG per mil you've seen with your media panel?
Like I mentioned earlier, around 1 × 10^11.
That's a different serotype, probably. Yeah, Gino and I talked about different
and different license procedures and stuff like that. Some of them as well. Yeah.
It all depends, you know, the plasmid DNA, the serotype, and we have some collaboration work. We could not show their data, so we got, you know, a higher with the collaborator work.
Thank you. I believe I have a question for you, Monica. So I'll ask you, and then you tell me if it's not for you.
Sure.
You mentioned manufacturing as overtaking clinical as a bottleneck. What do you view as key opportunities to increase AAV manufacturing capacity? Which steps in the process offer the most room for improvement in terms of increasing yield?
I don't think that's a question for me. It may be a question for Chao Yan or Gino.
I agree the moment I was reading.
Gino, would you like to go first? Or Natasha,
Gino?
Can you emphasize the question?
I will ask again, yes.
Yeah.
You mentioned manufacturing as overtaking clinical as a bottleneck. What do you view as key opportunities to increase AAV manufacturing capacity?
For me, I think, right now is already changed the workflow from adherent to suspension. So our suspension system definitely gave the opportunity to increase the scale. And I think right now is just, we could do the further optimization, reduce the DNA usage, reduce the cost, and produce more kind of repeatable, reproducible, the good quality thing. And for the mammalian suspension, without SV40 antigen, reduce the regulatory concern for the clinical. I think we are on that track. That's what my understanding.
Thank you, Chao Yan. Gina, would you like to comment?
I think just trying to increase the VG per cell, basically increase the productivity as well as help increase the cell densities that these things can be transfected at would help increase the yields.
Although our system is transient, we would like customers stay with our transient, this production workflow. I feel like eventually maybe stable cell line, is just like a further boost to meet the high VG per mil requirement.
Thank you, Chao Yan. Thank you, Gino. Our next question, I believe, is for you, Chao Yan. It is, how much boost drop did you see if you don't use the AAV-MAX enhancer?
Half. So if you use AAV-MAX enhancer, you double the VG per mil. And you keep the same infectivity. If you calculate the infectivity as VG per unit, so it's almost equivalent. You do not really negatively impact the virus, the function, and you just like double the VG per mil.
Thank you, Chao Yan. We have one more question for you. The question is, you mentioned that you achieved 32% full particles in your purification results. Was the 32% full achieved without AEX?
Without what?
AEX.
Oh, that's AEX. We have the empty full ratio result with multiple different, the analytical method, the AEX, AUC, and also the TEM, the image, and even the cryo-EM, all give us, relatively similar results, about the middle of 30%. But we need to do more serotype and to check more serotypes to confirm. And right now we are in the process to further, to get the idea, how about the DNA ratio? As I mentioned, the 3' DNA, the ratio, the harvest, the time, the kinetic, and the impact, the empty full ratio. We are in that process to try to get more information from more different serotypes.
Thank you, Chao Yan. It looks like we have no more questions for our great speaker. So I will conclude this Q&A session. And this also concludes this workshop. thank you to all of our speakers for delivering a very informative presentations. And thank you, our audience, for attending and participating in this workshop. Have a great day, afternoon, or evening, wherever you are. Goodbye.
Thank you.
Thank you.